Okay. So today class we are discussing about the RBC life cycle. Okay. So the lifespan of the RBC is 120 days. So after the 120 days, the aged RBC, they are passed through the capillaries present in the spleen and liver. So the capillaries in the spleen and liver are very small. That's why by passing through the capillaries in the spleen and liver, the rupturing of the RBC cells is occurred. So the aged RBC cells, the plasma membrane is fragile. That means what? Easily breakable. So aged RBC cells, their plasma membrane is easily breakable. So in that condition, they passed through the capillaries present in the spleen and the liver, they become ruptured because the capillaries in the spleen and liver are very small. So ఈ small pores నుంచి pass అయ్యేటప్పుడు the membrane of the RBC and the cells are becomes ruptured. So like this, the degradation of the by this, the degradation of the RBC is occurred. So after 120 days, how the degradation is occurred and how the RBC cells are again developed. This is the process of RBC life cycle. So how to degrade and how to reappear. But the RBC is not able to synthesize their cells. So the cells of the RBC are synthesized only from the bone marrow. That's why after destruction, after 120 days, after aging, these cells are first destructed in the spleen and liver by the capillaries present in the spleen and liver. Then they undergoes the phagocytosis. Here in the image, the red blood cells, the number 1 mark. See the number 1 mark. So this is the macrophages of this area is pink colored area is macrophage in the spleen, liver and the red bone marrow. So by passing through the spleen and liver capillaries, the rupturing of the RBC cells are occurred. After that, these RBC cells are digested by the macrophages present in the spleen and liver. So the digestion process is called phagocytosis. So the macrophages present in the spleen and liver digest the destructed RBC. So further, the RBC is separated into heme and globulin molecules. Total RBC ఎన్ని ఉంటాయండి? Total of 280 millions of the hemoglobin are present in a single RBC cell. Okay, so like that, after rupturing of the RBC cells by the spleen and liver capillaries, then there are further digestion processes occurred in the macrophages of the spleen and liver. Okay, after that, this digestion causes the separation of the hemoglobin molecule into the heme and globin molecule. So the globin, it is a nothing but polypeptide chain. That is the long chain of amino acids. So in the globin, these amino acids are released and they are further reused for the production of the proteins. So firstly, the macrophages causes the phagocytosis of the destructed RBC cells. Then they are divided into globin molecules and heme molecules. So these globin molecules, these are the nothing but long chain of the amino acids. It is a polypeptide chain. It is a long chain of amino acids. These amino acids, released amino acids from the globin, they are also reusable for the protein formation. Okay, so that's in that form, these globin destructed RBC cells, the globin molecules are reused. Then come to the heme molecule. Heme means what? In the heme, it consists of what? ఏముంటుందండి heme లో చెప్పండి. Middle of the heme consists of middle of the
iron molecules.
Yes, iron molecules are present. So from the heme, some part is iron part. Heme releases the iron portion and non-iron portion. So the iron portion of the heme, here two ga divide ayindi chudandi heme portion. It is the iron portion and non-iron portion. So the iron portion of the heme, it is a ferrous molecule. This is Fe3 plus. It is combined with the or binded with the transferrin in the circulation. So a heme molecule divided into iron portion and non-iron portion. So the iron portion is nothing but ferrous molecule. This ferrous molecule is binded with the transferrin in the circulation. So in the circulation, the iron molecule is present in the form of Fe3 plus transferrin. So it is forms a binding reaction with the transferrin. So this transferrin, it is a plasma protein present in the circulation. So after division of the heme into iron and non-iron, this iron portion is come into the circulation and binded to the plasma protein, that is nothing but transferrin. So and forms a complex with the transferrin. Ferrous and transferrin complex is formed. Okay. So this ferrous and transferrin complex, then it is enters into the muscle fibers, liver, and also in the macrophages of the spleen. So in the now the transferrin plus ferrous complex is in the blood. After that, it enters into the different regions like liver, the macrophages of the liver and spleen, muscle fibers. ఎక్కడెక్కడికి వెళ్తుందండి? ఈ బ్లడ్ సర్క్యులేషన్ నుంచి ఎక్కడికి రీచ్ అవుతుంది? Macrophages of the liver and spleen and also to the muscle fibers. Okay, so in the liver, it consists of a iron storage protein that is called ferritin. In the liver, it consists of a iron storage protein that is called ferritin. So circulation lo ఏ కాంప్లెక్స్ ఫామ్ అయింది చెప్పండి? సర్క్యులేషన్ లో ఏ కాంప్లెక్స్ ఫామ్ అయిందండి?
Ferrous transferrin complex.
Yes, ferrous and transferrin complex is formed. Transferrin complex is formed in the circulation. Then it is enters into the some of the organs like macrophages of the liver and spleen and also to the muscle fibers. Actually, already the liver is consist of a iron storage protein that is called ferritin. Okay, so these ferrous molecules in the circulation, it is in the form of ferrous plus transferrin. Then this complex is broken and then this ferrous molecule is combined with the ferritin. So storage purpose kosam this ferrous molecule is combined with the ferritin. This ferritin is nothing but a iron storage protein. So up to now what happens? Firstly, by the macrophages of the liver and spleen, firstly the capillaries of the liver and spleen are very small. That's why whenever the aged RBC cells are passed through the spleen and liver, they become ruptured. After rupturing of the RBC cells, they are become digested by the macrophages present in the spleen and liver, and they separated into globin and heme group. This globin, it is a polypeptide chain, then it is released in the form of amino acids. These amino acids are also reused for the further protein formation or for the protein synthesis. And also along with the globin, heme also released from the RBC cells. This heme, it consists of iron portion and non-iron portion. The iron portion is nothing but ferrous. It is comes to the circulation. In the circulation, a plasma protein, that is transferrin, is present. This ferrous forms a complex with the transferrin present in the circulation. After that, it enters into the some of the organs like macrophages of the liver, spleen, and the muscle fibers. Already the liver has a iron storage protein that is called ferritin. So these ferrous molecules present, ferrous and transferrin complex present in the circulation, then it is broken and these ferrous molecules are comes and form a complex with the ferritin in the liver. Now the ferrous molecule or the iron molecule is in the storage form. Whenever the need is there, it is released and helpful for the formation of the again RBC cells. So now in the liver, this heme group, that is iron portion of the heme, it is now in the form of ferrous plus ferritin complex. A complex in form and storage condition lo a complex in form chesni
Ferrous plus ferritin.
as ferrous plus ferritin complex is formed during the storage condition. After that, again, for the usage, for the helpful for the further synthesis of the RBC, for further reusable process, these ferrous molecules are further reusable for the metabolic processes. These ferrous molecules are separated from the ferritin, again forms a complex with the, comes in the circulation, again forms a complex with the transferrin, that is nothing but plasma protein. So in the circulation, the ferrous compound is forms a complex with the transferrin molecule. So always in the circulation, the iron portion of the heme is in the form of ferrous plus transferrin complex. Okay, during the storage conditions only it is present in the form of ferrous plus ferritin. So for the reusable process, again this complex is cleaved and the ferrous molecule is again come into the circulation for the release for the usage. So this forms a complex with the plasma protein present in the circulation, that is nothing but transferrin. So it forms the ferrous plus transferrin complex, then it enters the red bone marrow. So this complex is carried to the red bone marrow for the release of the RBC cells. Okay, so how it helpful? So by the endocytosis process, these ferrous molecules are enters into the endocytosis process is helpful for the allowing of the ferrous molecules into the this red bone marrow. After that, the red bone marrow is takes the amino acids or used the amino acids for the formation of globin, and also vitamin B12 is also used for the, it is nothing but cyanocobalamin. It is also helpful for the production of the blood cells, and also it used the erythropoietin. Erythropoietin, it is a hormone. This hormone is act on the mainly on the precursor cell of the RBC, that is nothing but erythrocytoblast. So like that, it forms a complex with the all these compounds and produced the undergoes the erythropoiesis mechanism in the red bone marrow for the release of the red blood cells. So what is the erythropoiesis? Erythropoiesis ante entandi?
Production of RBC
Formation
cells.
of RBC.
Yes, the formation of the RBC is called the erythropoiesis. So for the formation of the RBC, so degrade అయిపోయినాయి అదే కంటెంట్ లో బ్లడ్ లోకి cells అనేవి రీప్లేస్ అవ్వాలి. For that process, the erythropoiesis process is occurred in the red bone marrow. For that sake, it takes the ferrous molecules from the circulation into the by the endocytosis process from circulation into red bone marrow, the ferrous molecules are entered. Then for the synthesis of globin portion, it needs the amino acids. So these stored amino acids for the protein synthesis, they are also used for the formation of the hemoglobin. And vitamin B12 is also helpful for the formation of this hemoglobin. And also the erythropoietin, it is a hormone. It acts on the precursor cell, that is nothing but the proerythroblast. Then these activate the production of the red blood cells are nothing but erythrocytes. So this is the normal mechanism. After the produced red blood cells are enters into the circulation, and then this process is again continued. After 120 days, again the rupturing and phagocytosis occurs. This all the process is repeated. This is nothing but RBC cycle. So we are talking about only the iron portion of the heme, and there is a non-iron portion of the heme is also present. This non-iron portion of the heme, it is converted into biliverdin. This biliverdin, it is a green colored pigment. So this non-iron portion is converted into biliverdin. This is a green color pigment. This is further converted into bilirubin. This bilirubin is a yellowish orange colored pigment. ఏ పిగ్మెంట్ అయితే ఉంటుందో ఆ కలర్ లోకి ఇక్కడ మనకి మెన్షన్ చేశారండి. హీమ్ నాన్ ఐరన్ పోర్షన్ ఈస్ నథింగ్ బట్ ఏ biliverdin. It is a green color pigment. Then it converted into bilirubin. This bilirubin, it is a yellowish orange colored pigment. Then this bilirubin is enters into the liver and also into the small intestine. Okay, so it enters into the liver as well as into the small intestine. So now this enters into the liver. This bilirubin is converted into the bile. It enters into the liver. This bilirubin is enters into the liver and it is converted into the bile. This bile is stored in the gallbladder. Okay, so the bilirubin then enters into the liver. In the liver, by the help of liver cells, it converted into the bile. This bile is stored in the gallbladder. Whereas this bilirubin is also enters into the small intestine. From the small intestine to it reaches the large intestine. So in the large intestine, there is a large number of the bacteria are present. These bacteria, so from the small intestine, this bilirubin is enters into the large intestine. In the large intestine, there is a large number of the bacteria are present. These bacteria convert the bilirubin into urobilinogen. Bilirubin into urobilinogen. This urobilinogen is converted into two forms. One form is that is urobilin, other form is stercobilin. This bilirubin is converted into two forms. One is urobilin and other one is stercobilin. So this urobilin is enters into the kidney and eliminated in the form of urine. Then this stercobilin, it is a clay colored or brown colored pigment. This stercobilin is eliminated in the form of feces. It gives the color to the phases. Okay, so this is about the non-iron pigment. So the heme group really consists of iron and non-iron portion. The non-iron portion of the heme group is in the form of biliverdin. This biliverdin, it is a green colored pigment. Then it converted into the bilirubin. This bilirubin is enters into the liver and also to the small intestine. In the liver, this bilirubin is by the help of liver cells is converted into bile. This bile is stored in the gallbladder. Whereas in the small intestine, this bilirubin is entered, then it reached the large intestine. This large intestine is having the bacteria. This bacteria convert the bilirubin into urobilinogen. This urobilinogen is further converted into two forms. One is urobilin, another one in stercobilin. This urobilin is enters the kidney and released in the excreted in the form of urine. This stercobilin, it is a brown color pigment. It is excreted in the form of feces and it is also gives the color to the feces. So this is about the how the RBC cycle is occurred totally. So firstly, by the blood capillaries, the blood capillaries in the spleen and liver are very small. That's why these RBC cells are become ruptured by the capillaries present in the spleen and the liver, and then they undergoes the phagocytosis by the macrophages present in the spleen and liver. They are broken into two groups, heme and globin group. This globin, it is nothing but a polypeptide chain. That's why it is converted into the amino acids. These amino acids are used for the protein synthesis because the proteins are nothing but the long chain of the amino acids. Then it also releases the heme group. This heme group, it consists of iron portion and the non-iron portion. So this iron portion is enter the circulation. In the circulation, there is a plasma protein transferrin is present. So this iron portion is forms a complex with the transferrin, that is ferrous transferrin complex. Then after that, from the circulation, it enters into the some of the organs like macrophages of the liver or to the liver cells, or the macrophages of the spleen, or to the muscle fibers. So in the liver, it consists of a iron storage protein that is nothing but ferritin. So this circulation, ferrous transferrin complex is broken. The ferrous molecule is come and forms a complex with ferritin in the liver. Now the ferrous molecule or the iron portion of the heme is in the storage form. Whenever the need is there, it released again into the circulation for the further purpose. So this ferrous molecule is again enter the circulation and forms the complex with the plasma protein that is nothing but transferrin. Then it enters the red bone marrow for the purpose of production of the red blood cells. So this ferrous molecule enters the red bone marrow by the endocytosis process. After that, this red bone marrow is also need the amino acids for the formation of globin molecule and also vitamin B12 for the production of the red blood cells. This erythropoietin is hormone. It is a hormone. It is also needed for the activation of the precursor cell that is nothing but proerythroblast for the production of the red blood cells. So this hemopoiesis is a process of production of the red blood cells. It is occurred in the red bone marrow. Then these red blood cells are released into the blood for the further purpose. So then this heme is also consist of non-iron portion. This non-iron portion of the heme is called biliverdin. This biliverdin is further converted into bilirubin and it enters into the two organs one is liver, other one is the small intestine. In the liver, this bilirubin is a yellowish green or yellowish orange pigment. It is converted into bile by the liver cells, and then this bile is stored in the